Hey, good morning again, everyone, over here in the Whatcom Family Farmers room. We are going to get started right now. So if you're looking for those pesticide credits, get in here ASAP. Um, and yeah, we are going to probably run a couple minutes into lunch, but we'll get to those sign-in sheets ASAP once we're done with our presentations. So we're going to get started here with Savannah Phipps, who's presenting Developing Insights on the Use of Modern Breeding Tools for Assessing Root Lesion Nematode, Pratolynchus penetrans, Resistance in Red Raspberry. Take it away, Savannah.
Thank you for that lovely introduction, and thank you again for the invitation to come back again this year to provide an update on this project I've been working on. And if you've attended the other nematode sections the last couple of days, it seems like it's pretty grim in some sectors of management, but hopefully I can provide some hope moving forward into the future for managing root lesion nematode in raspberry production. I'm a PhD candidate at Oregon State University, but I have the opportunity to work with USDA scientists, Drs. Inga Zazada, who's the nematologist at the unit in Corvallis, and then the caneberry breeder, Michael Hartigan. They're both part of the same units as Jerry Weiland, if you saw his talk yesterday. So as an overview for this presentation, I will very briefly describe some of the red raspberry production in this region, the review of root lesion nematode life habits, and then review some of the employed and investigated methods. I will not belabor these points, so you won't have a whole repeat of what Tom Forge and Tom Walters has covered so far. Before we get into what my current research is on in investigating resistance and or tolerance. So if you're growing red raspberries in the Pacific Northwest, they do excellently here. But what makes this region so well suited for raspberry production? And that is the temperate maritime climate, the well-draining soils, and the sandy loam soils. And if you saw Dr. Griffin-LaHue's Deidre's presentation yesterday, we have this disease triangle where if you have a right environment and you have the right host, the only thing left is the pathogen. And as you might have guessed, the root lesion nematode also does really well here in those conditions. It's a significant soil-borne pathogen. As a reminder, it infects over 400 different plant hosts, so it's really hard to manage. It's also microscopic, so we're unable to see it unassisted with the naked eye. We have to use a microscope to see it. And not only is it a challenge here, but it's a challenge for red raspberry productions in Scotland, England, and Canada as well. And these areas also have very similar environmental conditions to us, so it makes a lot of sense. To better understand how we might manage this nematode, we can look further into its life cycle. As it has been previously mentioned, it's a migratory endoparasite, which means it doesn't have a portion of its life cycle where it just stops moving. It's continuously moving throughout the roots in the soil, back and forth. Sometimes they don't ever leave the roots. Sometimes they don't ever enter the roots. But it's this movement through the roots that causes the damage, right? And it has 6 life stages like other nematodes. We have the egg stage, 4 juvenile stages, and the adult stage. And it's when we get into the second stage, juvenile stage, where the damage starts occurring. The first juvenile stage stays within the egg. And, if you're stuck in an egg, you can't really feed on a root. So we're not concerned at that point in time. But once it emerges from that egg and it starts moving around, until it dies, it's an issue. Unlike other Pratolynchus species, this is one of the few species that does sexually reproduce. So you do have to have a male and a female nematode to have 4 nematodes. Other species are parthenogenic, so the female doesn't need a male to reproduce, but that's not the case here. Because it's one of the few species that requires a male, this is a diagnostic feature for this particular species as well. So we have the males, we get more Pratolynchus penetrans, we're going to have a greater proliferation of that population. And as I mentioned, that feeding within the roots really damages the vascular system, and that's what causes the symptoms that we see here. So below ground, we get necrotic root lesions, which is what gives its nematode its common name, the root lesion nematode. And because it's feeding and destroying those vascular systems, it's reducing those systems as well. But we're often seeing the above ground symptoms first. We're not really, looking into roots and being like, oh yeah, that's a problem there. We're seeing problems above ground. And we're seeing stunting, chlorosis, In more severe cases, it's reduced growth and fruit yields. And then in most severe cases, complete plant death. And it's further emphasized that this has been an issue in production for many decades. Like McElroy in 1977 details some significant biomass reduction losses. We're seeing a 66% there. Yield reduction was almost 47%. And most recently, in Zazada et al.'s 2015 paper, they're still seeing a significant biomass reduction, even though they're looking at more cultivars. And even more recently, Dr. DeVetter details in the 2018 paper that there's a 12% yield reduction. It's not as significant as the 1977 reduction, but we don't want to see any yield losses, period. So many decades since these papers, we've been looking at different management methods. We've looked at more conventional methods like soil fumigation and crop rotation, and even with the big brains that are involved on projects like these, alternative methods like biofumigation and solarization. And if you attended Tom Walters and Tom Forge's talk yesterday, this is kind of how the current state looks. The only thing that's really effective for reducing Pitylochus penetrans in populations is soil fumigation. All these other methods are either ineffective at reducing those populations or are currently not cost effective for growers like you to use. But you might notice that plant genetic resistance has this question mark. There are currently 4 papers looking at genetic resistance, and they come from the 80s. And technology since then has improved significantly. But from those papers, we can gather that there does seem to be resistance and tolerance present in the regional programs. So there's hope that we can monopolize on that to improve the IPM management systems here. And as a result, if we can get resistant and or tolerant varieties out to you guys, we can improve and make the IPM management plan for this nematode more sustainable. So how am I doing that? I have a really large field trial at the Washington State University Northwestern Washington Research and Extension Center. This is a field trial that encompasses a diversity panel featuring plants out of the USDA Oregon State University Breeding Program, the Washington State University Breeding Program, the British Columbia Berry Cultivar Incorporation Program, and select cultivars from national and international programs out of the National Clonal Germplasm Repository. And we've replicated this diversity panel 3 times in the field to encompass some of the soil variation that we know from this field. And furthermore, we have a paired treatment system where in the field, we have rows that are not inoculated with a nematode and then rows that are inoculated. And what I mean by paired is that, like, for instance, if this first plant was a meeker, the first plant in the inoculated treatment would also be a meeker. And like if the second plant was a willamette, both plants second in those rows would both be willamettes. So when you're walking through this field, you can make direct comparisons as well. And then how are we evaluating for that nematode stress? Over the course of these decades, researchers have looked at a variety of traits. We can quantify nematodes, which seems the most obvious, but that requires root sampling and then extracting those nematodes and counting those nematodes manually under the scope. We can also record biomass, whether that's above ground biomass or below ground biomass or the entire plant. They've also investigated cane diameter, cane height, cane number, and many more traits. But at the end of the day, they found that the most significant traits that are correlated to resistance or tolerance is nematode quantification and above ground biomass. So for nematode quantification, we're using a modified Behrman funnel method. Because the nematode is migratory, it allows us to easily extract it. These samples we're collecting, a golf ball size amount of roots. They sit in a mist chamber for 5 days. And while the roots are getting misted, as the nematodes are moving around, they're falling with the water and as a result falling to the bottom of the tube because they are denser than water. And then once after those 5 days, we collect the 20 mils off the bottom of these tubes and then manually count them under the scope. Because we only have so much space, this is approximately a 6-week process for the 2,000 samples that we get out of this field. And it's also time limiting because we have to manually count these and almost everyone in the lab is working on it. For the above ground biomass, we are recording the fresh weight above ground biomass. These plants are pruned to the crown at the end of the season when they're starting to go dormant. And in the 2022 season, they're pretty small, so we're able to use a tabletop weighing system where we cut them down and brought them out of the field to record on the table. But in 2023 and 2024, you can see that this plant's almost the size of me. And as a result, we developed a hanging arm system that allowed us to weigh the biomass infield as we progressed through it. I have finished collecting data for the last 3 seasons, and this will be the data that we will have on this trial. So for 2023 and 2024, we have nematode densities for then. We don't have the 2022 densities because we established this field pretty late in season. It went into the ground June 2022, and by the time we were cutting those plants down, we were worried that if we also dug up their roots, we would kill all of our hard work with putting them in. But we can see that there are relatively low densities in the non-inoculated plots throughout 2023 and 2024, which is good. We did do a Vitae application both these years to ensure that any native populations stayed low and that any effects that we were seeing in densities or biomass were not because of that. But we do see some pockets of the nematodes establishing themselves in 2023 and 2024. Even more so in 2024. We speculate that we have this hotter pocket in the eastern part of the field because there is a water cistern there, and soil moisture can contribute to a faster reproductive life cycle. And in 2024, they seem to have established more evenly throughout the field, as we might expect. In 2023, here are best linear unbiased predictors. They're kind of a modified mean that allows us to account for field variation as well as missing data, and it makes this data a little more interpretable. You might see here that we do have some higher averages for the inoculated over the non-inoculated. For the above ground biomass, we're still parsing out what exactly this means. I have looked at the raw values for this, and we're still seeing this trend as well. So let's continue a further moment of investigation for me. I don't expect you to read these genotypes because I'll get to those in this slide. And I've selected the highest 10 biomass values and then the 10 lowest. And we would expect between the 10 highest biomass values and the 10 lowest biomass values that we might see some resistance occurring here. We would expect more of a tolerance if we were seeing high biomass values with high nematode values as well. But for simplicity and time today, I'm only looking at what might be potentially resistant. So here, we have some differences occurring between the 10 highest biomass values and the 10 lowest nematode density values. But the one that is consistent between the two is WSU-1415. Now, before you go talk to Wendy about this, it is a breeding parent. It is not a selection. It's a cross between Meeker and Rubus sumatranus. So it's a wild Asian variety that is not cultivated here. And infield, it is a very tall, bushy, thorny plant that is not pleasant to work with at all. So it's not ready for cultivation at this point in time. But moving forward might serve as an interesting parent in the breeding program for incorporating resistance. But since this is only from 2023, we're really interested in seeing what 2024 looks like for this plant. I also included Meeker and Wakefield here as references for those selections and cultivars that are present in this table. I do have the 20 for 24 results, but I haven't gotten to the point where I have the blood values for them yet. And I'm hoping to have those in the next few weeks. There's only so much statistics you can do in a day before your computer gets a little too hot. But I'm hoping to have those values for you guys next year. And as you might realize, this is a ton of physical labor, and it's a super destructive process. And for breeders, it's not feasible to cut your plants down every year because you can't evaluate for fruit yields or fruit quality either. So we thought that we might be able to implement a different mode of phenotyping while still getting important data out. In other systems, we see that high throughput phenotyping using imaging can be successful in detecting nematode stress and then nematode resistance and tolerance in plants. But this has not yet been tested in raspberries. So the last couple of years, I've been conducting imaging using a mobile device, an iPhone 14 with the AgriPoint Capture app, and then an aerial device. In this image, it is a little drone that gets flown over the field, and I have a bunch of images, and hopefully we can correlate those. And it seems to be that this is super promising so far. So for the 2023 mobile imaging, there appears to be a similarly strong correlation between the number of points for a plant that's generated and then the biomass that was recorded. And then in the aerial imaging from 2022, the pixel count for those plants is also similarly strongly correlated with the biomass that was recorded. But you might notice that there's only a few points for the mobile imaging. And that is because there is a ton of back-end processing that needs to occur. So AgriPoint is conducting that processing that gives me the data back, but you might notice that their algorithm is still learning how to identify raspberry plants correctly. So in some instances, it's calling one plant as two plants, or two plants as one plant, and some additional reprocessing is needed to have the most accurate values. So the values in the last slide really show you only the ones that came out properly. But I'm hoping that we get more points as I get those results back. Additionally, the drone image processing is still taking some additional time. The drone is not just taking one giant field picture. As it's flying over, it's taking a bunch of little pictures, so I get the best resolution. And that requires stitching these images together to produce the image here. But you also might notice that our soil is not lavender, and it's not yellow or blue either. So there's some artifacts that is occurring from the imaging process that requires correction as well before we get to the final analysis, and that will still take some time. But once all of that phenotyping data is back, we can use the DNA data from these plants and the trait data and plug those into statistical models like the fixed and random model circulating probability to then detect significances between the genetics and the phenotypes that we're seeing to better identify what areas of the region we can incorporate into the breeding program. And furthermore, we can take those results and plug them into another statistical pipeline that allows us to select on individuals without phenotyping because we have that previous phenotyping data. And then with that established genetic data, you can draw on that for interpretations of the later downstream plants as well. To validate that these models are working as they should be, we'll be using a Monte Carlo cross-validation to verify that they're accurate. As you can tell, there's still a lot of work that's left in the pipeline, even though I do have all of that data. I have to finish processing that data before we can conduct that genome-wide association to detect what genetics are present there, and then also develop those models for being able to select on individuals that are resistant or tolerant. And then also, furthermore, evaluating the effectiveness of those phenotyping technologies for future phenotyping and even detecting nematode stress in your fields. But hopefully, at the end of the day, we can deploy these methods in breeding programs to improve the management of this nematode for the future. In summary, it's a significant soil-borne pathogen for red raspberry production here in the Pacific Northwest, and our current management relies heavily on chemical methods. There's no suitable alternative control at the moment, but this ongoing collaborative effort between the Pacific Northwest breeding programs and the nematologists to identify resistance can improve the availability of resistance for growers like you in the future. And this presents as the first study to evaluate plant stress in nematodes using these modern tools and technologies in raspberry as well. There's a lot of different groups and agencies involved on this project. I have a lot of scientists listed here, but it doesn't include all of the field technicians, the undergraduate interns, the other graduate students who have dedicated a ton of time and effort in helping me in the field and in the lab. And if I didn't have their help, I would still be there today. So I'm forever in debt to them. And this is also possible because of the funding received by the Northwest Center for Small Fruits Research and the North American Raspberry and Blackberry Association. And if there's time, I'm happy to take questions.
Awesome. Thanks, Savannah. Looks like we have a question. I'm coming at you with the mic now.
I guess the next step, are you doing genetic and now, mapping the raspberry gene?
Yeah.
And then figuring out where the resistance or susceptibility is, and can you use CRISPR to do something?
Well, That's probably beyond my PhD using CRISPR, and I know they have talked about CRISPR for other disease resistance in the past. I don't know if that's going to be a possibility here. I know there's concerns from the growers in using gene editing tools and technologies, but it'll probably be more classical breeding with identifying those areas and then determining like if we only have a few genes, then we can use one parent potentially to incorporate that more rapidly. But if we have multiple, it might take a little longer.
Awesome. Anyone else got questions for Savannah?
Hi, thank you for your research. A question about nematodes not related to the genetics of the plants.
Sure.
Are there any predators of the nematodes that affect raspberries? And has there been any research done in relation to that?
In regards to predators, I'm not entirely certain if there's any other predatory nematodes that feed on this one or fungi. I have seen a couple papers that have looked at bacterial sort of controls. Those papers are back from the 90s, and considering that I haven't seen any new papers regarding those species moving forward, it doesn't seem that they had success with employing those in the field. But I think it is an excellent area of research to further expand upon. I don't think we totally understand the network quite yet with this nematode and the other nematodes around it.
Great answer. Great questions. Anyone else? We have a time for one more. If anyone has any other questions for Savannah while we have her. Awesome. Well, thank you again, Savannah. We really appreciate it.